Imagine having the ability to rewrite the very blueprint of life itself, to correct genetic errors, enhance desirable traits, and even eradicate diseases at their core. Gene editing allows us to make precise changes to an organism's DNA, the intricate code that dictates everything from our appearance to our susceptibility to diseases. This groundbreaking technology holds the potential to address some of humanity's most pressing challenges, from inherited diseases to global food security. By understanding the mechanisms of gene editing, we can begin to grasp its profound implications for the future of medicine, agriculture, and even the environment. Imagine our DNA as a long, winding instruction manual, with each gene acting as a specific set of instructions. Gene editing tools act like molecular scissors, allowing scientists to target and modify these instructions with remarkable precision. This could involve correcting a genetic defect that causes a disease, inserting a beneficial gene, or even switching off a harmful one. Join me as we unlock the secrets of gene editing and explore its capacity to reshape our world. Among the various gene editing tools, CRISPR-Cas9 stands out as a true game changer. This revolutionary technology has democratized gene editing, making it faster, cheaper, and more accessible than ever before. CRISPR-Cas9 is essentially a two-part system that evolved in bacteria as a defense mechanism against viruses. The first part, CRISPR, acts like a cellular library, storing snippets of viral DNA that the bacteria has encountered before. The second part, Cas9, is a protein that acts like a pair of molecular scissors, guided by the CRISPR library to precisely cut and edit DNA sequences. By engineering the guide RNA, researchers can direct the Cas9 protein to target and cut virtually any desired DNA sequence. The simplicity, versatility, and affordability of CRISPR-Cas9 have made it the go-to tool for gene editing, revolutionizing fields ranging from medicine to agriculture. While CRISPR-Cas9 has taken the gene editing world by storm, it's not the only tool in the toolbox. Scientists have developed other gene editing techniques, each with its own strengths and limitations. One such technique is zinc finger nucleases, or ZFNs, which were among the first gene editing tools to be developed. Another approach, transcription activator-like effector nucleases, or TALENs, offers a similar mechanism, utilizing engineered proteins to target and modify specific DNA sequences. Although ZFNs and TALENs paved the way for gene editing, they are more complex and expensive to engineer compared to CRISPR-Cas9. The diversity of gene editing tools available underscores the rapid pace of innovation in this field. The transformative potential of gene editing extends far beyond the laboratory, promising groundbreaking applications in various fields, including medicine, agriculture, and environmental conservation. In medicine, gene editing offers hope for treating and even curing genetic diseases that have plagued humanity for centuries. By correcting faulty genes, scientists could potentially treat diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease at their root cause. Moreover, gene editing holds immense promise for cancer research, allowing scientists to engineer immune cells to target and destroy cancerous cells with greater precision. Beyond treating diseases, gene editing could revolutionize agriculture by enhancing crop yields, improving nutritional content, and increasing resistance to pests and diseases. Imagine crops that can thrive in harsh environments, require less water and fertilizer, and provide higher yields, addressing global food security concerns. Similarly, gene editing could lead to healthier and more sustainable livestock, resistant to diseases and producing higher quality products. Environmental conservation efforts could also benefit from gene editing, such as controlling invasive species and enhancing the resilience of coral reefs facing climate change. Section 5. The Ethical Frontier: Navigating the Uncharted Waters. The unprecedented power of gene editing comes with profound ethical considerations. One of the most pressing ethical concerns is the potential for unintended consequences. Altering one gene could have unforeseen ripple effects on other genes or even the entire organism. The potential for exacerbating existing social inequalities is another ethical concern. Access to gene editing therapies and enhancements could be unequally distributed, potentially widening the gap between the wealthy and the disadvantaged. Moreover, the very notion of manipulating the human germline, the genes passed down to future generations raises profound ethical questions. Open dialogue, transparency, and careful consideration of diverse perspectives are paramount as we chart a responsible course for the future of gene editing. Section 6. Designer babies. Separating fact from fiction. The concept of designer babies, genetically modified children with desirable traits, often dominates public discourse surrounding gene editing. It's important to distinguish between two types of gene editing, somatic and germline editing. Somatic gene editing targets cells in the body that are not involved in reproduction. Changes made through somatic gene editing are not passed down to future generations. Germline editing, on the other hand, involves modifying genes in sperm, eggs, or embryos, resulting in changes that are inherited by future generations. Currently, most gene editing research focuses on somatic gene editing for treating diseases in individuals. Open discussions and a focus on ethical guidelines are essential to ensure that gene editing is used responsibly and for the benefit of humanity. Section 7. Drawing the lines, the evolving landscape of gene editing regulations. As gene editing technology advances, governments and regulatory bodies worldwide are grappling with the challenge of establishing ethical and legal frameworks. Many countries have implemented strict regulations governing gene editing, particularly germline editing. Some nations prohibit germline editing altogether, while others allow it only for research purposes under stringent oversight. The development of international guidelines and agreements is crucial to address the global implications of gene editing. Organizations like the World Health Organization, or WHO, are actively involved in facilitating discussions and developing ethical frameworks. Open dialogue, collaboration, and a commitment to transparency are essential to ensure that gene editing technologies are developed and deployed responsibly. The ongoing evolution of gene editing regulations reflects the dynamic nature of this rapidly advancing field. Section 8. A future shaped by gene editing. Gene editing has the potential to reshape our world in profound and lasting ways, offering solutions to some of humanity's most pressing challenges. The future of medicine is likely to be transformed by gene editing. We can envision a future where genetic diseases are a thing of the past, cured at their root by precise gene editing therapies. Beyond treating diseases, gene editing could enhance human capabilities, leading to a new era of human potential. Agriculture is poised for a gene editing revolution, leading to a future of sustainable and abundant food production. Imagine crops engineered to withstand harsh climates, resist pests and diseases, and produce higher yields, addressing global food security concerns. As we navigate the ethical complexities and harness the transformative power of gene editing, we have a unique opportunity to shape a future where scientific progress goes hand in hand with human well-being. Section 9. Navigating the potential of gene editing, a balancing act. The future of gene editing hinges on our ability to navigate its immense potential while addressing the ethical concerns it raises. Open and transparent communication is paramount. Scientists, policymakers, and the public must engage in ongoing discussions about the potential benefits and risks of gene editing. Responsible research practices are essential. Scientists must conduct gene editing research with the utmost integrity, adhering to rigorous ethical guidelines. Equitable access to the benefits of gene editing is crucial. By embracing a balanced approach that fosters both innovation and responsibility, we can harness the transformative power of gene editing to create a healthier, more sustainable, and more equitable future for all. Section 10. A call for responsibility, steering gene editing towards a brighter future. The gene editing revolution is upon us, and with it comes a profound responsibility. We hold in our hands a technology with the potential to reshape life as we know it. But this power must be wielded with caution, guided by ethical principles and a deep respect for the sanctity of life. We must engage in open and honest dialogue about the potential benefits and risks of gene editing. We must establish robust regulatory frameworks that foster innovation while safeguarding against unintended consequences. And we must ensure that the benefits of gene editing are shared equitably, bridging the gap between the haves and the have-nots. By embracing a spirit of responsibility, collaboration, and a deep respect for the sanctity of life, we can steer gene editing towards a brighter future for all of humanity.